I would like to introduce Dr. Stephanie Sherman. Dr. Sherman obtained her PhD in human genetics from Indiana University, Purdue University, and Indianapolis. She trained as a genetic epidemiologist and is currently a professor in the Department of Human Genetics at Emory University in Atlanta. She has focused her research on identifying genetic and environmental factors that explain the severity of clinical conditions associated with Down syndrome. She and her colleagues across the U.S. have established DS360, a clinical research program to accelerate research in Down syndrome, with the overreaching goal to find treatment targets, reduce the effect of the extra chromosome 21, and increase the quality of life for those with Down syndrome. Welcome, Dr. Sherman.
Yeah, perfect. Thank you. Thank you for inviting me to come, and Stephanie for all the work that she's done, and the Down Syndrome Indiana group for organizing this. I have to say, though, Vince just leaned over and said to me, "You're going to follow him?" It's like I had been thinking that the whole time. Oh, my goodness, that was just so inspiring. I think the one thing I wanted you to take away and using the words of Carl is, I hope that we can move or accelerate research from the slowness of pond water, which I had never heard before. I love this, being slow as pond water, to hopefully, the rapids in a river. So that's what we're here for today, is to really explain to you how your participation and how our interaction together as clinicians and researchers might move that research to a faster pace. So I'm going to just kind of give an overview of what you'll hear today. And I'm going to try to put that in the context of the NIH Down Syndrome Research Plan that Vince mentioned. So what's happening in the world of Down syndrome? I first need to acknowledge all the work that the different foundations have been doing. This is a collaboration. This is the only way we'll move forward rapidly. And so I'll I won't define anybody specifically, but you can see there's a lot of organizations that take part, as well as organizations like Down Syndrome of Indiana. Okay, so what's our common goal? It's to increase the quality of life of those with Down syndrome, try to decrease the health effects of that extra chromosome 21. Now, why is this such a problem? Why have we moved somewhat slowly? This is not a change in a single gene that, some of the many conditions that we work with in medical genetics. This is really the increased dosage of many genes on one chromosome. So it's a bigger problem to try to deal with, and we don't want to specifically dissect out each gene. We know that they all work together and really lead to some alterations of biological pathways that we want to define. In order to do that, again, it's a collaboration. But your needs as families and individuals with Down syndrome, it's your needs that guide the research priorities. So, and then it's also your participation in these projects combined with the new research tools that help reach the goals that I mentioned. So let me go over some of the research tools, just to give you an idea. This is going to be quick and not comprehensive, but just to give you an idea of some of the more current tools. One thing that you'll hear about today are model systems. Model systems are just taking some other organism or some other type of experimental system, and these are critical in understanding the pathophysiology, or what's really going on with the disease and how the disease progresses without essentially harming the individual with Down syndrome. That we know we can't use those individuals in some of the experiments that are necessary. Here I've just shown what we call an ideogram of chromosome 21 and how it's kind of defined scientifically. But what I want to say is one of the biggest tools that we use in model systems is the mouse model. The mouse model actually carries the genes that are comparable to chromosome 21 on 3 of its chromosomes listed here. So you can use the mouse model, but it's complicated. You have to try to put it all together to make sure that mouse model is a good model of trisomy 21. In the corner here is, I want to recognize Muriel Davidson, who was a woman that kind of devoted her career to creating these mouse models, knowing that this would be an important tool. So here's one of the first mouse models created, the TS65DN. You'll hear that term potentially when you read about research. But what I want to show is now there's many new mouse models. And each one of those different models has a different repertoire of the chromosome 21 gene. So you can begin to dissect out which are the important genes on chromosome 21, for example, that potentiate the risk of childhood leukemia or the congenital heart defects. So these tools are very important. A new tool also is induced pluripotent stem cells. So why is this important? This is a new technology where you can take some cells from an individual with Down syndrome or any other medical disorder. You can take those cells, say perhaps skin cells, you can put them in a dish, and essentially you culture them to bring them back to a naive state, to a state where they're pluripotent. In other words, they can move into any other kind of tissue. And that's important because every tissue regulates its genes in a different way. So, for example, you can take those pluripotent cells and push them to be a neuron-like cell, or push them to be a cardiomyocyte or a heart cell. And then you've got the tools to say, okay, what drugs ameliorate some of the effects of trisomy 21? And so it's a very fascinating tool that we can now use. And it can be involved in, for example, disease modeling, as I was saying. You can actually use these cells to say, if I give these cells this particular compound or drug, does it help ameliorate the symptoms that I see? So again, very potent type of tool that has become available in our research. I want to emphasize that the research goals are an iterative, or reaching those goals is an iterative process. So we work with individuals with Down syndrome and their families to understand what's going on, what the questions are important to ask, how they look, what for example, here is showing this EEGs that you can do now with little ones and really see what's going on in the brain, especially with the activity, for example, associated with seizures. You can take that back into the model systems. You can see how do you change that effect. For example, in an iPS cell, if you give it that drug, do you reduce that excitatory transmission? And then you have to bring it back again, because if you find something out, you have to go back to the individual with Down syndrome and say, is this a potential? Is this drug a potential? What am I thinking? What are the other background genes that might lead to response or no response of this gene? So it's a very iterative interaction. Okay, so those are just some new tools, and you'll hear more today of other tools. But I want to talk about now some of the talks that you'll hear today and put them in the context of the NIH research plan on Down syndrome. This plan that was developed really brought together the experts in the field, the clinicians in the field, the individuals with Down syndrome, their families, and said, what are the needs? What are the priorities? How do we want to spend our money to best help reach goals? So this is the list of the main headings of it, and we'll go through each one of these. So how about pathophysiology and disease progression? So this is an iterative process, so there's many tools you can use. Model systems help you understand some of these aspects, for example. How do you characterize what's going on? This is just a picture of a pre-neuron and post-neuron diagram with a synapse between the two, which is a very important active area of transmitting information from one neuron to another in that synapse. So you can ask in model systems, what is the synaptic function? How is it altered? The vesicles that carry the neurotransmitters, how are they moving through those neurons? Are they doing it properly? Are they releasing the neurotransmitters properly? Are the spines of the neurons, do they look proper? Again, if you add a drug, will they change? Will they morph into a more mature-looking neuron? Or, for example, you can ask questions, and we know that APP, which is a very important gene involved in the risk for Alzheimer's in individuals with Down syndrome, is one of the genes that's in a trisomic state. But you can ask in these model systems, given it's in an increased dosage, how does it change that environment of the neuron, or how does it change the environment of the brain? And so that's APP processing, and how does it change the signaling pathways? These are just some examples of things that you want to learn related to the pathophysiology and disease progression. You can also complement those model studies, as I said, with clinical studies. So I'm giving the example of a study that we're involved in called the Down Syndrome Cognition Study. And so what we're doing is we're asking the questions, let's characterize an individual or a population of individuals with Down syndrome in terms of their cognitive and behavioral function. And then let's find those factors that explain that extreme variation. Why are some individuals with Down syndrome profoundly affected while others have very mild outcomes related to that trisomy 21? So the whole goal of that, again, is to characterize the whole distribution, the whole spectrum of impairment that is seen among individuals. Again, low performers, high performers, identify the environmental factors, the genetic factors that might explain that variation. From that, you can start to identify the biological pathways that are involved in that, and then find target interventions to rebalance what's going on. So that's the strategy in this Down Syndrome Cognition project, as well as other projects. And today we'll also hear from Dr. Chanel and Han, who are from the University of Illinois at Urbana-Champaign, about the progression of outcomes from infancy to adolescence. So that will be coming up. The next component of the NIH plan is related conditions that are more frequent among individuals with Down syndrome compared to the general population. And thinking about how do you screen for them in terms of prevention, how do you diagnose them, what are the functional measures that are best used to characterize those conditions. So, for example, a very active area of research is how to refine and develop specific outcome measures related to cognition and behavior. How do you characterize them so they're sensitive enough that you can actually identify change in clinical trials? A very important part of the field. And there's many people involved. Some of you might be involved in Dr. Edgin's project to characterize these kinds of outcomes in very young children, or David Hessel's, who's at the Mind, looking at different tools that are encouraged by NIH to use to characterize cognition and behavior. Or maybe some of you are involved in Len Abadudo's studies that involve language and how to best characterize language in its full spectrum. Measures of dementia, depression, autism, sometimes they're very hard to characterize on the background of intellectual disabilities. Again, a very active area. And then there's new technologies of imaging that can help identify biomarkers that might be involved in the prediction of onset or progression of particular disorders like dementia. Also, Down syndrome and aging, a very important field. And when you think about this, this is wonderful. I mean, the average age now of individuals with Down syndrome is around 60. That has progressed so much over the last years. So now it's important to understand, what does aging look like in an individual with Down syndrome? That wasn't an issue before. Before, it was trying to get individuals with Down syndrome through the perinatal period, right, with some of the medical conditions. Those advances have been made now. But now it's important to focus on aging and what does that look like. So identify factors associated with, for example, the onset of and the progression of dementia. Another example is, when do you start hormone therapy for adult women with Down syndrome? That's something that hasn't been addressed and is very important. How do you implement the general screening, health screening and prevention measures that we do for everybody in this room. But how do you implement that, or what are the guidelines? There are guidelines set up, but make sure they're implemented for adults with Down syndrome. Cholesterol testing, just all DEXA scanning for bone density. You want to make sure those are all in place. What are the gaps? What do we need to improve in terms of care for adults with Down syndrome? And then what are their social needs? Not only of the individual themselves, the adult, but also the families to help support them, or the support system in general. And then, of course, treatment. How can we ameliorate some of the effects, the aging effects that we see? And then Drs. Head and Schmidt from the University of Kentucky later on will be talking about this very topic. They have a very active program in aging in people with Down syndrome. Okay, treatment and management. And this is treatment, as I think Vince brought up. What we're trying to do is ameliorate the effects of trisomy 21 at all levels. There's co-occurring conditions that definitely are targets of intervention: leukemia, congenital heart defects, gastrointestinal problems, and then the list goes on. But very important things. Now we can treat the symptoms, or we can do the corrective surgery on some of the birth defects, but how do we start to think about prevention and reducing the outcomes? Co-occurring psychiatric and neurobehavior, here I've listed autism, ADHD, depression, regression. These are very important things that reduce the quality of life of individuals with Down syndrome. How do we treat those? And as I mentioned before, age-associated disorders and cognition and daily life functioning. So we're going to hear Dr. Roper from here at IUPUI is going to talk about treatment in trisomy 21 and really talk about it from a scientific perspective. In addition, Dr. Martin, again from Indiana, the University of Indiana, is going to talk about some of the benefits of using orthosis in young children with Down syndrome. So caring for some of the outcomes that interfere with daily living. And then in terms of cognition in daily life, we'll have two examples of projects there. Dr. Prena from Indiana University looking at the potential of using video games in improving memory and learning. And then from Dr. Swinford, again here at IUPUI, kind of a new way of looking at helping individuals live, laugh, and dance. I love the title. And so connecting the mind, body, and soul. So that's something to look forward to. And lastly, research infrastructure. Again, NIH was very clear about having to establish the research infrastructure for aspects of conducting research, making each site research ready, making each site a potential clinical trial site if potential therapeutics come to the forefront. And again, Vince talked a bit about the Down Syndrome Connect, the NIH registry for individuals with Down syndrome, and how this can be used to expand research participation, and how this Down syndrome, the DS Connect, can be used to begin to understand the natural history of individuals from infancy, individuals with Down syndrome from infancy through their lifetime. So we'll hear from Dr. Kaiser about Down Syndrome Connect. And then the thing that we in the scientific community are very keen on is finding resources that are shareable. This is such a key part. We each are involved in our own research, but we want to see whatever we can do to take our research products or our research findings and share them in the whole community. So that's what we're promoting as the Down Syndrome 360. We think of it as a critical program to accelerate and facilitate research in Down syndrome. And I've shown here the same type of pathway that we're trying to improve on to end up thinking about how to develop the best interventions to target the imbalance caused by trisomy 21. So what does this mean? This is just a small scale that can grow, but what we're aiming for now, just to get the foundation started, is a collaborative effort to just say we'll take 100 participants, we'll characterize them very carefully in terms of their outcome measures. What are the birth defects that the individual has? What are the cognitive level of that person, etc. And then just do 1,000 genomes. Take the blood from each one of those individuals, or saliva, whatever is the potential. Characterize the genome and have that as a shareable piece of data. Do it once. Just do it once, do the whole genome, and it can be used in many different ways and being used responsibly. I want to make sure that's understood. So just to begin, again, to lie the foundation through the, for example, the Down syndrome cognition study and a Down syndrome heart study that we've been involved in, there's about the different sites here that we have for recruitment, and each one of these sites, I could say, is research ready. So they know how to conduct research. They're very involved in it themselves. What we're trying to do right at this point as a proof of principle is working with the London Down Syndrome Project, a huge project in the UK that has studies from infancy through adulthood. And our studies, the Down syndrome cognition study, overlap significantly with their adolescent studies. So we're finding the tools to bring these data together to integrate them so that others can use those data. And the Trisomy 21 Research Society, again, an international society, is helping with us to characterize or to help us define which clinical assessments should be done. Biobanks are important. This, again, is to take a sample, process it, and have it in a true bank so that others can use that. It can be inventoried. We know how to use it well and responsibly for people who ask for access. And then lastly, as I said, to take the genetic data or any of the omics data, I'm sure you've heard that term, any of those data, put them in a data repository so that it can be shared with other researchers that have good ideas that they can then grab onto those data and move ahead rapidly instead of starting the whole process over again. So it's an exciting time. I want to say that we are today recruiting or enrolling people for Down Syndrome 360. If they're interested, we can take biological samples at this setting. Tracy Rosser, who is right there, and Elizabeth Sablon is right next to her, are involved in that process and can explain more and answer your questions. So with that, I wanted to say, again, our common goal: increase the quality of life with individuals with Down syndrome, decrease the health effects of the extra trisomy 21. And I want to emphasize, though, we're very focused on helping individuals with Down syndrome. But everything we learn from individuals with Down syndrome in terms of these co-occurring conditions actually can be applied to those without Down syndrome who have those same conditions, like congenital heart defects. So everything that's done in this room and done in your society really helps not just those in the community, but much further out. So thank you very much.